Voice of the Sea, learning from experts across the ocean.
Welcome to Voice of the Sea.
In this special episode, we're taking a look at mercury in the ocean food web. We'll go from the research vessel Kilo Moana, where fishes are caught and initial mercury tests are performed, to the labs of UH Mānoa, where scientists study trace mercury from the samples collected at sea. We start off with Dr. Brian Popp from the University of Hawai, School of Ocean and Earth Science and Technology. Brian explains what mercury is and the tools he uses to identify both the concentration and the source of mercury in our food fishes. Good morning, Brian. It's great to be here. I'm excited to learn about your work studying mercury and how it accumulates in the food web in the ocean.
Excellent. Thanks for coming.
I'm particularly interested because I like to eat fish, and I know that mercury accumulates, and so the fish that I like to eat might have a lot of mercury, and there's some personal relevance there, and I want to hear your story.
It started a while ago, and we had just some preliminary observations just from other people's data that mercury concentrations in fish not only change as a function of where they feed within a food web, but also at what depth they feed. And we found that the same size fish at the same trophic position, or the same position within that food web, had higher concentrations of mercury. Different species have sort of preferred depth ranges that they feed in. And so when you think about ahi, there are a couple of different species that we sell as ahi. One of them is yellowfin tuna and the other big-eye tuna.
Right.
Big-eye tuna feed deeper in the water and they have higher mercury concentrations, even though they eat the same sort of things that yellowfin tuna eat, but they eat them in a deeper. And they seem to accumulate more mercury. Mercury has many different masses. There are 7 different masses of mercury.
What does it mean if mercury has different masses?
They weigh different amounts. There are mercury that have low mass and high mass, and they react differently in reactions.
So that means I could have the same amount of mercury in 2 samples, and one would be heavier than the other, even though they look to be the same amount.
Yeah.
Okay.
Yeah. And it can be used as a fingerprint of where the mercury comes from. So an isotope nerd like myself and our colleagues at University of Michigan, having that many isotopes is like nirvana in a nerdy way. But some of the isotopes, particularly the odd isotopes, can react in a slightly different fashion when they're exposed to sunlight. These so-called photochemical reactions in a laboratory setting, when you have mercury compounds and expose them to intense sunlight, they degrade, and they do so with this mercury isotope fingerprint. So what is really cool about this in terms of the ocean is that we have a fingerprint for mercury that has undergone chemical reactions in sunlight. And where does that happen? Well, it's near the surface. We found that, those that feed near the surface have this fingerprint of photochemical degradation. And as our work evolved, it became clear that the reason why fish near the surface appear to have lower mercury concentrations is that they're feeding in an environment where the organic mercury that is taken up is degraded by sunlight. And those that feed deep, it's not exposed to sunlight as much. We still see a bit of a fingerprint because mercury gets transported to deeper depths.
Does that mean that that photo degradation actually broke the mercury down and it's no longer mercury?
No, it just means that it turned it back into elemental mercury that doesn't get bioaccumulated to the same extent.
I see.
The form of mercury that gets bioaccumulated in fish and in us is something called monomethyl mercury. And the monomethyl mercury, in terms of our health, it doesn't matter what the masses are, and it doesn't matter if the isotopes are distributed in a mass-dependent or mass-independent way. It's still methyl mercury. If you are concerned with mercury in your diet, the fish that you should eat that have the lowest mercury are those that feed near the surface. So an important finding is that we appear to have used mercury isotopes as a fingerprint for understanding why fish that feed at the surface are lower in mercury concentrations.
Next, researchers Joel Bloom and Laura Mota from the University of Michigan explain their role studying the transfer of mercury in the ocean's food web.
My name is Joel Bloom. I'm a professor in the Department of Earth and Environmental Sciences at the University of Michigan. And I'm here on the Kilo Moana studying the cycling of mercury around the planet, and in particular in the Pacific Ocean. I've been working on understanding the chemistry of mercury on the Earth's surface for the last 15 or 20 years. And my research has taken me many different places in the world. Most of that has been done on land. And this is my first oceanographic cruise, and I'm out here sampling and studying mercury in the Pacific Ocean and in the marine food webs here. Mercury has a very complicated environmental chemistry to it. It changes forms. It's released in one place on the globe and will move around the globe over long distances. And there's a lot of interest in trying to understand how mercury is getting into fish here in the Pacific Ocean and other oceans. And that's really the ultimate goal of my project here. I'm working with Jeff Drazen and Brian Popp, and together we've been studying mercury starting in the atmosphere. The mercury that is a gas comes across the ocean in the atmosphere, largely from the combustion of coal in Asia. It's deposited in rainfall into the surface ocean, and then it undergoes a whole series of chemical transformations and ultimately enters into the marine food web. Then works its way up the food web, is bioaccumulated, getting to higher and higher concentrations until it gets into top predator fish, where it can reach levels that are hazardous for people to consume large amounts of fish. So we want to understand this whole process in more detail. I'm here with a graduate student, Laura Mota, and together we're doing a whole series of measurements and experiments of mercury throughout this whole, what we call biogeochemical system, in order to understand this process better.
My name is Laura Mota, and I'm here from University of Michigan, where I'm currently doing my PhD in geochemistry. We are in the Kilo Moana at Station Aloha, which is about 8 hours from Hawaii. And I'm here doing some photochemistry experiments in mercury. We're collecting samples to do mercury isotopes. So all these different conditions is to, one, learn how the signature is happening at the pathway, and two, learn more about mercury chemistry. Because for many, many years, people have been studying metal mercury, and nobody really has a clear idea how photochemistry happens. So here we have some mercury 2 and metal mercury with a couple dark controls to make sure that whatever we see with the light is not happening by some background reaction.
The University of Hawaii's Sea Grant College program focused on Hawaii's coasts and its communities through sustainable development, safe seafood supply, sustainable coastal tourism, hazard resilience, and healthy coastal ecosystems. Hawaii's Sea Grant.
Welcome back. University of Hawaii professor Brian Popp shows us some of the lab equipment his team uses to understand where in the environment fish are feeding. Brian uses samples of fish tissue to trace elements like carbon and nitrogen.
We prepare that sample with a bunch of chemistry, and it's a couple-day procedure to form a liquid that contains the different amino acids in there. And what we'll do is we'll draw up a little bit of that liquid, only like a couple of microliters of it. And we need to separate those amino acids because in this liquid, this 1 microliter of liquid, we're going to have maybe 15 or 20 different amino acids. And we want to measure the isotopic composition of each of those so we can use them to understand something about the trophic level of the organisms that we prepare that sample from. So what we do to separate it is that we use something called a gas chromatograph. And that gas chromatograph is hooked up to Lady Gaga here. Lady Gaga only cares about the nitrogen gas that goes in, but the amino acids don't have nitrogen gas. So we have to separate those amino acids and then turn them into a gas that the mass spectrometer can measure. So what we do is we take that sample and we inject it here. So we'll take and we'll put this in and we'll shoot in our sample. And where that goes is it goes into something called a gas chromatographic column. This column is 60 meters long, but it's hollow. It's like a tube. And that tube, the inside diameter, is only 1/3 of a millimeter. You can maybe fit about 4 or 5 of your hairs in there, but not many more. That kind of brownish coating is an organic film that the compounds interact with. So the amino acids are sliding through this tube, and the little amino acids slide through there more rapidly than the big ones. And it gives us our way to separate those. They end up into this little tiny combustion oven. And that combustion oven is at 980 degrees. And at 980 degrees in that oven, it converts that amino acid to carbon dioxide and nitrogen gas, which then travels through more of those tubes, but not as an amino acid, as a gas. And they go through this, our little interface here. And the interface just allows us to direct where those gases go. There are some gases that we want eventually to end up going into the mass spectrometer, but there are other things that we don't. And this is our way to divert any of the gases that we don't want in there.
And how does the mass spectrometer work?
The mass spectrometer takes the nitrogen gas, the molecules kind of go into what's called an ion source. And the ion source is the source of ions. The nitrogen gas gets bombarded by electrons and it knocks an electron off, so it creates a positively charged molecule. And once it's positively charged, we can accelerate it out of that ion source. So this ion source makes molecules. It also, there's 10,000 volts applied to it. And if you have a positively charged molecule that's sitting in 10,000 volts, it doesn't like to sit there, so it gets shot out. Yeah. But what this does, it creates those ions, it accelerates those ions, and it also allows us to focus those ions. And then those gases, those ions, ionized gases, go into this. This is a tube with a magnet around it. And what the magnet does is it affects those masses differently, such that it's where we separate our masses. The heavy masses, because it's curved like that, it'll go through the magnetic field and the heavy masses will be diverted, but the lighter masses will be diverted a lot more because they're lighter. They then get focused into something that detects those ions, and we can measure a ratio from those, the ratio of the nitrogen gas containing the heavy isotopes to the nitrogen gas containing the light isotopes. So the mass spectrometer creates the ions, it separates them, and eventually allows us to measure a ratio. So all of this is sort of one instrument, but the mass spectrometer is a little bit different, and this is kind of a preparation system. And we can put any kind of a preparation device we want onto a mass spectrometer.
So what direction are you going to take your research next?
We want to see if the mercury cycle that we've been able to interpret from the isotopic compositions of mercury, we want to apply our model, our hypothesis, to different regions. And our hypothesis would be in a productive region that has lower water clarity, we should see less degradation of mercury through photochemical degradation. And the mercury concentrations may be higher in the algae and in the fish that we find there.
The Curriculum Research and Development Group in the College of Education at the University of Hawai at Manoa. CRDG's training roots go back over 40 years. Through professional development programs, curriculum workshops, research on teaching methodology, individualized school and district training, and so much more. The Curriculum Research and Development Group, improving schools, improving education. CRDG.
Welcome back. We're talking with oceanographer Brian Popp and geologist Christian Miller. By sampling different water depths and comparing them to the metal isotopes that are found in the tissue of fish, Brian's work can build an understanding of how metals are accumulated in the fish that people like to eat. We start off on board the research vessel Kilo Moana on one of Brian's research trips, where a crew of scientists and technicians go to sea for weeks at a time to study the ocean. On board, Brian is going to show us his mobile clean lab.
You've now just entered our trace metal clean bubble. We erected this plastic tent to keep all the metals out. We have a hood here that filters all the particles out, and it gives us a flow of really, really clean air over these filters that keeps this room nice and clean. Let me show you some of the equipment that we use to sample water. We get it out of its protective bag. This is called a GoFlow bottle. It's different from the Niskin bottles that we saw outside in that the opening to it on the top and bottom are made so that it stays closed when it goes through the water. You can see when this spins, there's an opening there, and every 90 degrees it alternates between open and closed. The reason why we want this bottle closed when we send it over the side and into the water is that the very top surface of the water has a lot of contaminants in it, especially metals. We're on a metal ship, and there's metals that are always being eroded off that ship. It's like measuring iron in seawater is very important, and we're on this iron ship. It's a ship made of steel. So we don't want that contamination in our bottle. So we use this special bottle. It's a little bit more difficult to use than the 24 bottles on the rosette outside, but it stays clean as it goes through the air-water interface and the upper 30 feet of the ocean. We know how deep the bottle goes, and by sending what's called a messenger down, it's a coded weight on that line. It comes down, it hits this, and voila, closes the bottle, and we get our sample. And we can then bring the line back up with the bottle and take this GoFlow bottle into the laboratory and sample it under these clean conditions that we've created with our trace metal free bubble. We're sampling mercury in this case, and it's mercury that's dissolved in seawater. We're interested in the concentration of mercury as a function of depth because we've seen mercury changes in fish that feed at different depths within the ocean. And we want to know if that's what's driving the concentration gradient in fish, whether it's a concentration gradient in dissolved mercury. When we bring our samples back here too, we want to be really, really careful about any contamination from the environment. We have special clean labs that we use only for our mercury analyses. We don't have to worry about that for nitrogen and carbon. There's no contamination that we have that's a problem. But if mercury gets in from either from the dust on the floor or from fluorescent lights, there's a lot of mercury in fluorescent lights, that's a problem. So we have to use special clean lab to do that.
Cool. Can we take a look at your clean lab?
Sure.
All right. The Clean Lab was built in 2006 to provide an environment free of trace metals for researchers to study geology, volcanology, and the environmental effects of human activities.
You guys should, when you guys are ready to step over onto here, you can put your feet inside some of these. One thing to be aware of these is that the floor can be slippery when you're wearing them because there's very little grip. So there should be a pair for you. Thank you. And a pair for Kenisa.
Thank you. Then we just go through there.
Usually just put these on over your overhead.
Yeah. And just a quick question, have either of you been swimming this morning in the sea by any chance?
No.
Perfect, perfect. The reason I ask is because we do analyses of strontium in this lab, and strontium is the 5th most abundant cation in seawater. So it's a potentially large contaminant if you walk in here with sea salt on your skin and in your hair.
You look nice. Dr. Popp.
Why, thank you, Dr. Duncan.
We are gowned up. You know, the shoes are off. We got footwear that's worn only in this lab because obviously the bottoms of your feet and your shoes are very dirty. And the simple fact is we are covered up as much as possible because once we go inside, we are going to be the dirtiest things in the lab. We're going to be dirtier than the floor. We're going to be dirtier than the counters. And so as we come into the clean lab, we try and make things progressively cleaner. So, you know, there's a line past which we do not wear shoes. Then once we come in here, we always gown up and get ready in this room before heading in there. And we always be sure to step here on the sticky mat so that whatever particles are on the bottoms of our feet and of our booties get taken off, and so that we don't take them into the lab. Also, in every room in the lab, there will be one of these, which basically describes the air pressure. And the thing that makes the clean lab so clean and allows it to stay so clean is that the inside is at a higher air pressure than this room. So any dirt that does fall to the floor gets swept out by air moving underneath the door, and this room is higher in pressure than the one out there, and the one out there is higher than the hall. So as you go in farther and farther, it should get cleaner and cleaner.
Exploring Our Fluid Earth is a dynamic curriculum developed by the University of Hawaii's Curriculum Research and Development Group, teaching ocean science concepts through the disciplines of physics, chemistry, biology, and ecology. Exploring Our Fluid Earth is now available freely online. Find out more at exploringourfluidearth.org.